Welcome back to the Deep Dive. This is the show where we take that pile of sources you need to read, we do the work for you, and we give you the shortcut to being well-informed on some of the most critical topics in science and tech. And today our mission is, well, it's one of a kind. We are going deep on PROTON, a really groundbreaking graph AI system. It's designed for multiscale neurological disease modeling.
And we're not just talking about a theoretical model here. This is a 578 million parameter system trained on a massive custom-built knowledge graph called NeuroKG.
And the really extraordinary part, I think, is that they didn't just stop at the predictions. They took those predictions and validated them in the real world.
Oh, absolutely. It's a phenomenal piece of work. It really feels like a glimpse into the future of translational AI. We're looking at a system that can connect genes to cells, to environmental factors, to actual patient outcomes.
And then test those connections for Parkinson's, for bipolar disorder, and for Alzheimer's.
Three of the most devastating diseases we face.
And before we unpack this incredible blueprint for AI-driven discovery, we want to welcome you to Generative AI Futures, which is proudly brought to you by ByteGoose AI. So let's set the stage. Why does a system like PROTON even need to exist right now? I mean, we talk a lot about the challenge of neurological disease, but it helps to be reminded of the sheer scale of the problem.
The stakes are just immense. I mean, neurological disorders are the single leading global cause of disability. Full stop.
Not just one of them, the leading cause.
The leading cause. We're talking about an impact on over 3 billion people worldwide. It's responsible for more than 11 million deaths every single year. And yet, for most of these conditions, we just don't have disease-modifying treatments.
We're just managing symptoms.
Exactly. We're managing symptoms. We're not reversing the underlying cause. And that lack of progress is, a central bottleneck in all of modern medicine.
And the problem isn't a lack of effort, right? It's the brain itself. It's just unbelievably complex. A drug has to work on a protein inside a cell in a specific brain region without causing problems everywhere else.
Precisely. The disease mechanism, it spans all these different scales that we've historically studied in isolation. You have molecular biologists looking at one gene, but they might miss the context of the cell type where that gene matters.
Like the dopaminergic neuron in Parkinson's.
Right. And then you have clinicians looking at patient outcomes, but they miss the upstream molecular causes. The real problem is that the pathology is happening across all these levels at the same time.
So the integration is the innovation here.
That's the mission of PROTON, to bridge those scales, to offer a rational, predictable, and scalable way to do AI-driven discovery in neurology. It's about moving from these silos of data to a truly integrated understanding.
So our mission today is to walk you through that entire process step by step. We're going to start with the data foundation, then get into the transformer architecture, and then the really exciting part, the results from the lab and the clinic. Okay, so let's start at the very beginning. The foundation of this whole thing is the data structure. You said traditional methods, a simple database or a big spreadsheet, just don't work here. Why not? Why couldn't they just put everything in Excel?
Well, a spreadsheet is great at one thing, listing facts. It can tell you drug A treats disease B, or gene X interacts with protein Y, but it's terrible at capturing the context and, more importantly, the relationships between all those facts.
So it's about more than just columns and rows.
Oh, much more. In biology, and especially in the brain, everything is defined by connectivity. A protein's function depends on the pathway it's in, which interacts with other pathways, all inside a specific cell in a specific part of the brain. You need a data structure that treats those connections, those relationships, as just as important as the things they connect.
And that's the knowledge graph.
That is the knowledge graph. And in this case, it's NeuroKG. This is PROTON's brain, its data set. It's what's called a heterogeneous biomedical knowledge graph.
Okay, so heterogeneous, meaning it's made of many different types of things.
Exactly. Think of it like a giant subway map of the nervous system. The nodes are the stations, a gene, a cell, a disease, a drug, and the edges are the tracks connecting them, the relationships like regulates or is located in or interacts with.
And let's talk about the scale of this map, because this doesn't sound small.
Yeah.
How much data did they actually pull together to build this thing?
It was a massive effort. NeuroKG unifies 36 different biomedical data sets and ontologies. We're talking about everything from drug bank to protein interaction networks like ADRD to pathway databases like Reactome.
Okay, so what's the final count? How big is this graph?
The final graph has 147,020 unique nodes, stations on the map across 16 different types, and a total of 7,366,745 edges or connections across 47 different relationship types.
7.3 million connections. That's a universe of biological information. How did they actually link all these different types of data together, from molecules to patients?
They were very systematic about it. At the molecular level, you have the basics: genes, proteins, pathways. Then at the cellular and anatomical level, they grounded that information in specific cell types and brain regions using anatomical ontologies.
And then you get to the more applied stuff.
Right. Then you have the clinical level, which connects diseases, phenotypes, and even electronic health records. And then the external factors, the pharmacological and environmental level. So drugs, contraindications, and environmental exposures from databases like the CTD. The whole point is that a gene node can be connected to a drug node through a cell type node.
And this is where it gets really interesting, because they didn't just use generic biological data. They used single nucleus RNA sequencing, snRNA-seq, to make it hyper-focused on the brain. How do you even begin to integrate data from millions of individual brain cells?
That step is absolutely critical. It's what makes this a neurology graph, not just a general biology graph. A general graph might tell you a gene is active in the liver, which is useless for Parkinson's. So they integrated these snRNA-seq atlases to ground everything in the brain's cellular landscape.
And what was the scale of that cell data?
They started with data from over 3.7 million cells from the adult human brain. That's 2.4 million neurons and nearly 900,000 other cell types.
And I'm guessing they drilled down even further for the specific diseases they were studying.
Yes, they did. For Parkinson's, they integrated specific data from PD patients and controls, focusing on the substantia nigra, which is where those dopaminergic neurons die. We're talking about another 387,000 individual nuclei just for that.
So the graph can go from just this gene is in the brain to something incredibly specific.
Exactly. It could potentially say this gene is expressed differently in an astrocyte versus a dopaminergic neuron in the substantia nigra of a PD patient. That level of granularity is what gives it its predictive power.
It's what allows for that disease specificity.
Correct. And that whole process created entirely new nodes and edges in the graph for specific cell types and subtypes. They actually constructed over 160,000 new edges, just connecting cell subclusters to their top marker genes. It's a huge shift from a generic map to a neurology-focused one.
Okay, I have to ask about quality control. When you're pulling from 36 different sources and creating millions of new connections, how do you make sure you're not just integrating garbage?
A fundamentally important question, because when you integrate 36 sources, you also integrate all their mistakes and biases. So the researchers used a really clever state-of-the-art tool called PaperQA2.
Which is another AI.
It is. It's a generative literature search agent. It's been validated to perform at or even above the level of a human expert at finding and synthesizing evidence from biomedical papers.
So they used one AI to fact-check the foundation for another AI. That's pretty cool. How did it work?
They basically aimed PaperQA2 at the parts of the graph around the major neurological diseases and asked it to systematically check the edges. So for a given connection, does strong published evidence actually support this link?
And what was the verdict? How trustworthy is this foundation?
The results were very strong. PaperQA2 found that published evidence supported 83.87% of the key edges it tested.
So still, that means about 16% couldn't be confirmed.
Right, and that doesn't mean they're wrong necessarily. It could mean the connection is novel or only supported by weaker evidence. But the fact that they could transparently validate over 83% of the core relationships gives you immense confidence that NeuroKG is a robust, verified map of neurological biology.
Okay, so we have this gigantic high-fidelity map of the brain. Now we need the engine, the thing that can actually reason over the map to find hidden routes. And that engine is PROTON.
Right, and its full name is a 578 million parameter pre-trained relational transformer for neurology.
When most people hear transformer, they immediately think of things like ChatGPT. But this is different, right?
It's a whole different beast. LLMs are transformers trained on sequences of text, one word after another. Proton is a relational transformer. It's trained to reason over a network. It doesn't read sentences, it learns patterns of connectivity.
So if an LLM reads a book, what is Proton doing?
Proton is navigating that subway map we talked about. If you ask an LLM what treats bipolar disorder, it's recalling text it has seen. If you ask Proton, it's actually tracing the paths from the bipolar disorder node to all the drug nodes. And that path might go through a protein, through a specific cellular pathway. It's performing true relational reasoning.
And the specific architecture is called a heterogeneous graph transformer, or HGT. Why is that heterogeneous part so important for a graph like NeuroKG?
The heterogeneity is everything. I mean, NeuroKG has 16 different types of nodes and 47 types of edges. A simple, homogeneous graph model would treat them all the same, which would be a massive loss of information.
Can you give me an example of that?
Sure. Think about an edge between a drug and a disease. An edge-labeled indication means this drug helps, but an edge-labeled contraindication means this drug is harmful. A simple model might just see a link. The HGT is built to understand that nuance.
And how does the attention mechanism, the core of a transformer, work in a network?
Well, in any graph neural network, a node updates its information by getting messages from its neighbors. In a dense network, a gene might be connected to 1,000 other things: proteins, diseases, cells. A traditional model might just average all those messages. It's noisy and inefficient.
Just too much information coming in at once.
Exactly. The HGT's attention mechanism lets the model learn which neighbors are actually important. It calculates these attention scores to selectively focus on the most relevant connections when it's passing messages. It's learning to ignore the noise and amplify the important biological signals.
And you mentioned the different types of relationships. How does the model mathematically know the difference between treats and inhibits?
This is a really elegant part of the architecture. It uses what are called type-specific representation spaces. Essentially, for each of the 47 edge types, the model has a unique set of mathematical matrices. This forces the model to learn that, say, a drug disease link operates in a completely different conceptual space than a gene-gene link. It prevents all these distinct biological meanings from just collapsing into one meaningless vector.
Okay, that's the architecture. So what did they actually train it to do? What was the goal?
The goal was a self-supervised link prediction task. They basically gave the model the huge NeuroKG map, but with a bunch of connections hidden, and its job was to predict whether a connection should exist or not.
So it's trying to score real connections as high and fake connections as low.
Precisely. For every real, true edge in the graph, they created a corresponding fake negative edge. And the model's job is to learn the rules of neurological connectivity so it can tell the difference.
And did it work? Did it learn the rules?
It did, robustly. On a held-out test set of connections it had never seen before, PROTON achieved an area under the curve, or AROC, of 0.9145 and an accuracy of over 82%. That confirmed that this huge model had successfully learned the complex rules encoded in NeuroKG's 7.3 million edges.
I have to stop you there because this just sounds computationally massive. Training a 578 million parameter model on a graph with 147,000 nodes. How do they even manage that?
Yeah, this gets into a key problem in graph AI called the neighbor explosion. Since PROTON looks 3 layers deep, the final information for any single node depends on its neighbors' neighbors, and its neighbors' neighbors' neighbors.
So the amount of information it needs to process for just one node grows exponentially.
It explodes. In a dense network like this, the full message-flowing graph for even a small batch of nodes is way too big to fit into the memory of a GPU. You can't just load the whole map at once.
So what's the solution? How do you train it?
They used a very smart sampling strategy, a heterogeneous neighborhood sampling algorithm. So instead of trying to calculate everything at once, they trained the model in these small mini batches of 512 edges at a time. And for each batch, the algorithm intelligently subsamples the local neighborhood.
But wait, if you're just randomly sampling the neighborhood, don't you risk missing important information, especially from rarer nodes or less common connections?
That is the critical problem, and they had a brilliant solution for it. The sampling wasn't uniform. It was proportional to the square root of the relative node prevalence.
Okay, break that down for me. What does that mean?
It means it actively counteracts data bias. If you just sampled randomly, a very common node type, like a well-studied gene, would always get picked. A rare node type, like a specific cell subcluster, would almost never be seen. By sampling based on the square root of how common they are, you effectively upweight the rare nodes. You give them a much better chance of being included in the training process. It's a way of forcing the model to pay attention to the less studied, potentially more novel parts of the graph.
That's fascinating. So it's a computational trick to solve a biological data bias problem.
Yeah.
And the end result of all this is that every node in the graph gets its own vector representation or embedding.
Exactly. And they showed that these final embeddings were biologically meaningful. When they mapped them out, diseases clustered together based on clinical similarity, and drugs clustered together based on their chemical structure. It proved the model wasn't just crunching numbers, it was learning real biology.
Okay, so with that foundation in place, let's see this thing in action. Case study 1, Parkinson's disease. It's complex. It involves genetics, protein aggregation, environmental factors. A perfect test case.
Right. And the first goal was to see if PROTON could work as a computational proxy for really expensive lab experiments. So they ran 3 in silico screens, basically virtual experiments across the entire human proteome.
What were those 3 screens looking for?
The first screen ranked every protein-coding gene based on how likely it was to be genetically associated with Parkinson's risk. The second ranked every protein based on how likely it was to directly interact with alpha-synuclein, which is the protein that clumps up in PD.
And the third was a combination of both.
Yes, and that was the most powerful one, because a gene might be linked to PD risk but have nothing to do with the core pathology, or a protein might touch alpha-synuclein by chance. By looking for genes that had a strong signal in both genetic risk and mechanistic interaction, PROTON could zero in on the most biologically plausible targets.
And how do you validate something like that? A purely computational prediction?
You test it against the real world. They benchmarked PROTON's predictions against the results of 6 independent, previously published genome-wide experimental studies on alpha-synuclein.
And how did it do?
The results were definitive. The combined screen found the experimental hits in 5 of the 6 validation studies, and it consistently beat the standard baseline method, which is called random walk with restart in all 6. To give you some numbers, in one validation set, it achieved a normalized enrichment score, or NES, of 2.30. In another, using a different lab technique, the NES was 2.16. These are strong signals that PROTON's relational context is providing superior predictive power.
That's impressive for reproducing known science. But did it uncover anything new? They looked at this bigger question in Parkinson's. How do all these small effect genetic risk factors from GWAS studies actually lead to neuron death?
This is a huge gap in the field. So first, they got a list of 681 genes that are known to be essential for the survival of dopaminergic neurons. This came from a big CRISPR screen.
So a list of genes that, if you break them, the key neurons in Parkinson's die.
Yes. Then, PROTON ranked all other proteins in the network based on how likely they were to be connected to each of those 681 essential genes. And finally, they checked where known Parkinson's GWAS hits landed on those lists and compared that to where GWAS hits for 10 other diseases like Alzheimer's or asthma landed.
Ah, that's a clever design. It's asking if the connection between PD risk genes and DA survival genes is specific, or if it's just general biological crosstalk.
And it was highly specific. PROTON predicted the strongest links between the DA essential genes and the PD GWAS hits. The median rank was significantly better or stronger than for any of the other 10 diseases. It basically validated that the network wiring between PD genetic risk and the specific vulnerability of these neurons is unique.
And finally, the environmental piece. Pesticides are a known risk factor. How did PROTON try to predict which specific chemicals were the most dangerous?
They used a technique called few-shot learning. They fine-tuned the model on a tiny data set, just 28 pesticides known to be associated with Parkinson's and 100 known negatives. The model had to learn the network signature of a toxic pesticide from that small sample.
And the validation for this had to be based on actual toxicity, right?
Correct. They tested its predictions on a list of 696 pesticides, which included some that were already experimentally proven to be toxic to patient-derived dopaminergic neurons in a lab dish.
And what did it find?
The results were striking. The model ranked one of the most notorious organochlorine insecticides, endosulfan, at position 9. That put it in the top 1.29% of all 696 predictions. It was followed by 2 other known toxic pesticides. It proves the model can connect the dots from an environmental chemical through the network to a specific cellular toxicity outcome.
Okay, so let's shift gears from neurodegeneration to neuropsychiatry. Case study 2, bipolar disorder. This is a lifelong illness, and treatments like lithium don't work for everyone. The goal here was drug repurposing.
Yes, and this was a really tough test for the model. First, to prove it could reason from scratch, they trained it on a version of NeuroKG where they deliberately removed all known drug bipolar disorder connections.
So the model was completely blind. It didn't know lithium or anything else was used for BDD?
Completely blind. And even then, when asked to predict treatments, it managed to place 81.25% of the known drugs in the top 10% of its predictions. So it could rediscover existing therapies just by analyzing the network structure.
That's the computational proof. So after they trained it on the full graph, what was the novel drug candidate it proposed?
It nominated a few interesting compounds, but the one they chose to validate was calcitriol. That's the active form of vitamin D.
Vitamin D. That seems unexpected for bipolar disorder.
It is. I mean, there are some very loose epidemiological links between vitamin D and mood, but there was no strong mechanistic reason to think it would work. PROTON, however, predicted it with high confidence based on its network path.
And you can't just test a drug for a psychiatric disorder in a simple cell line. You need a better model. And they used patient-derived cortical organoids.
The mini brains, exactly. They grew these from stem cells derived from 5 BD patients and 4 healthy controls. It's a critical system because it preserves the patient's genetic background and allows you to study the disease in a 3D human brain-like structure.
And to measure the effect, they used deep proteomic profiling, looking at thousands of proteins at once.
Right. First, they established the baseline. The untreated organoids from the BD patients had 512 proteins that were expressed differently compared to the healthy controls. That was their disease signature.
So what happened when they added calcitriol, the AI's prediction?
This is the centerpiece result. After treating the BD organoids with calcitriol, they did an unsupervised analysis of the proteome, and they found that 4 out of the 5 calcitriol-treated patient samples clustered more closely with the healthy controls than they did with the untreated patient samples.
Wait, say that again? The drug shifted the entire molecular profile of the patient's cells back towards a healthy state.
A massive normalization of the disease signature. It's definitive proof that this non-obvious drug candidate picked by the AI fundamentally reversed the molecular pathology in a highly relevant patient-derived system.
So did the data give them any clues as to how it was working?
It did. It pointed to several pathways involved in metabolism, protein homeostasis, and synaptic function. But one of the most interesting findings was that the treatment seemed to reverse ADR overexpression.
And ADR is specifically linked to bipolar disorder, isn't it?
Yes, very strongly. ADAR proteins are involved in RNA editing, which is a process that can alter genetic information after it's been transcribed. This process is known to be dysregulated in BD. So the fact that calcitriol reversed this suggests a potential new mechanism of action.
And was it just acting like a weaker version of lithium?
No, and that's also great news. The analysis showed that its effects were distinct from lithium's. There was some overlap, but calcitriol seems to work through substantially different mechanisms, which means it could be a new therapeutic avenue, not just another version of an old one.
If the organoid validation was a high bar, the ultimate test has to be in actual patients. So for the final case study, Alzheimer's, they took PROTON's predictions and tested them against real-world clinical data.
This was the final frontier, moving from the lab bench to population-scale evidence. And they were very smart about it. They knew they couldn't just test 100 random predictions. They needed a rigorous filtering pipeline to get to the most plausible, testable hypotheses.
So what were the steps in that pipeline?
Step 1. PROTON screened all 8,160 drugs in its database and ranked them for a potential Alzheimer's indication. Step 2, and this is key for repurposing, they only looked at the top-ranked drugs rank less than 60 that were already FDA approved for something other than dementia.
So drugs that are already known to be safe and are being used by people right now.
Exactly. And step 3 was a reality check. They only kept the drugs that had been prescribed to over 1,000 patients in the Mass General Brigham healthcare system, because that's where their clinical data was coming from. They needed enough patients to have the statistical power to see an effect.
And how many candidates made it through all those filters?
Out of more than 8,000 to start, they were left with just 8 top drug candidates to validate.
So describe the validation cohort. This is where we move from a few organoids to hundreds of thousands of lives.
The scale is immense. It was a retrospective analysis using the electronic health records from 610,524 patients in the MGB system, tracked over a 7-year period. The question was simple. Did taking one of these 8 drugs reduce a person's risk of developing dementia?
But EHR research is notoriously tricky. How did they deal with the biggest problem, which is confounding? For example, the people taking a drug might be sicker or different in some way to begin with.
They were incredibly rigorous. For each of the 8 drugs, they created 2 perfectly matched groups. The treatment group patients who got the drug for its intended non-dementia reason, and the control group patients who had the exact same condition but weren't prescribed that specific drug.
Ah, so you're not comparing people with diabetes taking a drug to the general population. You're comparing people with diabetes taking the drug to other people with diabetes not taking the drug.
Precisely. It controls for that primary indication. And then they used advanced statistical methods. Cox proportional hazards models to estimate the risk over time, and a technique called inverse probability treatment weighting, or IPTW.
What does IPTW do?
It essentially creates two synthetic populations that are perfectly balanced on dozens of factors like age, sex, BMI, smoking status, other illnesses. It makes it so the only significant difference between the two groups is their exposure to the drug that PROTON predicted. It's the gold standard for this kind of analysis.
So after all of that, the graph, the AI, the filtering, the massive cohort, the advanced stats, what was the hit rate? Did the AI's predictions actually work?
They worked incredibly well. 5 of the 8 PROTON predicted drugs were associated with a statistically significant reduced risk of Alzheimer's and related dementias.
5 out of 8 in a clinical validation of this scale, that's a huge success.
It's a tremendous success. It shows the signal coming out of the AI was very, very clean.
And which drugs had the biggest protective effect?
The single strongest effect was for a drug called aflibercept. This is a drug used to treat wet age-related macular degeneration, an eye condition.
An eye drug.
An eye drug. And taking it was associated with a 37% reduction in dementia risk, a hazard ratio of 0.63.
That is just an unbelievable finding. A drug for the eye protecting the brain.
And it wasn't the only one. They also found strong protection from dapagliflozin, which is a drug for type 2 diabetes and kidney disease, and valsartan, a common blood pressure medication.
And what about the 3 that didn't work?
It's just as important that 3 of the drugs showed no significant effect. That tells you the AI wasn't just picking common drugs or finding random statistical noise. It was selectively identifying specific drugs with real neuroprotective properties based on their network biology.
So when you pull all of this together, what we're looking at is more than just a single discovery. It's a whole new process, right? The AI experiment discovery loop. What does this mean for the future?
It means we're moving beyond just building better models for the sake of it. Saying our model is 5% more accurate. This is about generating specific, high-confidence, testable hypotheses that can be immediately validated.
It collapses the timeline. What used to take decades of slow, serendipitous discovery could become a rational, predictable process.
That's the blueprint. It shows how you can couple AI directly to molecular systems, to cellular organoid systems, and to clinical EHR systems to just dramatically accelerate translational research. It shows, at scale, how all these different biological factors converge on a disease.
But we have to be critical. As impressive as this is, what are the limitations we need to keep in mind?
Of course. First is scope. They've tested 3 very common, well-studied diseases. The real test will be extending this to rare or understudied neurological conditions to make sure it's truly generalizable.
And the data itself, NeuroKG, is a reflection of what we already know. So it has to have biases.
That's the second point, data bias. Knowledge graphs are subject to things like publication bias. Well-studied genes have more connections. PROTON is trained on that bias, so it might tend to rediscover what we already know and overlook the truly novel, less connected targets.
Which is tied directly to the model's own tendencies, right?
Yes. Transformer models on graphs tend to favor these high-degree, well-connected nodes. They have more information flowing through them, so the model is more confident in its predictions about them.
So there's a risk that it makes it harder to find those really surprising low-degree connections that could be the real breakthroughs.
That is the fundamental tension. The researchers tried to mitigate it with that square root sampling we discussed, which helps a bit. But you still have this trade-off between prioritizing reliability, which favors the well-known stuff, and prioritizing novelty. That's a challenge for the entire field.
So what's the vision moving forward? How does this discovery loop evolve?
It has to be a living system. NeuroKG needs to be constantly updated with new data, new single-cell atlases, new proteomic studies, and the model needs to be continuously retrained. The ultimate goal is to create a pipeline where a computational prediction moves directly into a dedicated preclinical model or even an early-stage clinical trial designed specifically to test the AI's hypothesis.
Turning drug discovery from an art into more of a rational engineering discipline.
That's the path, a generalizable foundation model that generates high-value, testable hypotheses.
Well, that brings us to the end of our deep dive on PROTON. We've gone from the 7.3 million connections in NeuroKG to a 578 million parameter transformer, and through 3 absolutely stunning validation case studies.
In Parkinson's, it reproduced complex genetics and flagged a specific toxic insecticide. In bipolar disorder, its prediction, calcitriol, was shown to normalize the disease proteome in patient-derived mini-brains.
And in Alzheimer's, its hypotheses held up in the toughest environment of all. Over 610,000 patient records, where 5 of its 8 predictions showed a significant real-world protective effect against dementia. So let me leave you with a final thought on that. The research found that drugs for completely different conditions, like aflibercept for the eye, with that 37% reduction in dementia risk, or dapagliflozin for diabetes, had these massive protective effects on the brain.
So if AI can find these connections, protective effects for the brain hidden in drugs that target the eye or the kidney, what other profound biological overlaps are just waiting to be discovered across the entire human body? Things that our siloed way of doing research has completely missed.
And how much faster can we get life-saving treatments into clinical trials by testing these high-confidence, non-obvious connections that only an AI that can see the whole map could ever find? Something to think about until the next deep dive.
Thanks for joining us.
We'll see you next time.